Okay, so this is Engine Study Problem Set 3, Question 1, and this is my video on motor proteins. So part of our lecture, our guest lecture the other day, was on flagella and the composition and structure of the flagella. So one of the things that we learned is that flagellum have a 9x2 formation, which is basically these 9 kind of microtubule doublets on the outside, and then 2 little microtubules that run down the very middle of the flagella. So a lot, as we learned in class, a lot of different bacteria have different kind of flagellum formation. This picture, you can see that there's one single flagella coming off of the bacterium, but sometimes there's one at each end or kind of a lot of flagellum all over. During the guest lecture, we learned that a shear stress can be placed on the microtubule doublets within the flagellum, which causes the flagella to kind of bend, and then it can kind of beat back and forth in a sort of pseudo-sinusoidal manner, which causes the thrust that the bacteria would use to push itself through the medium that it exists in. Okay, so your question must be, how do vesicles travel through the flagella? And I haven't talked about vesicles yet, so it might be hard for you to ask that question, but never fear, we'll continue. So, motor proteins use ATP to pull vesicles along microtubules. So this is what this video is going to be about. So vesicles are essentially these little lipid bilayer filled things which have, they can have a lot of different things inside them, but they're usually like fluids like enzymes and stuff like that. So a flagella needs to carry these vesicles from the base of the flagella all the way to the end. So what it uses to do that is something called the motor protein. And I'm basically going to be going through what a motor protein is and what it does. So you can see in this little cartoon here that this vesicle is being carried along our microtubule, which we talked about in the last slide, from the base of the, basically the flagella, right where the flagella meets the bacteria, all the way to the end of the flagella. So this is a kind of a GIF of what we think this exactly looks like. This is a little bit too small to see with an optical microscope, and when you go down to a scanning electron microscope for biology, I think that you have to kill whatever it is and then basically cover it in like a gold powder or something like that to be able to observe it. But this is kind of another cartoon to describe what exactly we think this looks like. So it's kind of taking step by step, and you can see that it's holding on to the vesicle or the organelle that's filled with this enzyme, and this is the long microtubule or microtubule doublet that's running along the flagellum. Okay, so now this is a video. I'm going to go through how this works. Okay, so those are catalytic cores. There are 2 of them. There are 2 motor heads. Those are neck linkers, and there's 1 neck linker for each catalytic core. That is the microtubule doublet, and that's the organelle up there that's not shown. Those are coiled coils, and yeah, so as it's going to go through, there we go. Each head contains an ADP, which moves randomly, driven by Brownian motion, which is pretty cool. So they're just kind of randomly, Brownian motion kind of is causing them to move around in an unorganized way. Oh, and then one finds contact with the microtubule doublet, and it binds really tightly, which releases an ADP. So then an ATP comes in and takes its place. It enters at the binding site, which then gives the neck linker enough energy to zipper onto the catalytic core, throwing the second head forward, which is what's happening now, attaching it to the microtubule. So it whips it down with some force, and then that will in turn, subsequently, eventually release its ADP. There we go. And then ATP will take its spot. Then the other head, which just happened, hydrolyzes the ATP and releases phosphate. And you can see that this happens back and forth and back and forth. So one head will take in the ATP, and then basically the process will continue as these little guys take little steps across the microtubule, pulling the organelle or pulling the vesicle along the microtubule all the way from the base all the way to the tip, which is pretty neat. Okay, one thing I want to mention before I finish is that this has been happening for over 3 billion years, which is pretty wild because that is a really, really long time. And like you can think of like, okay, the dinosaurs happened like 65 million years ago, and humans have been around for, I don't know, 10,000 years. But this has been happening for 3 billion years. Like that's wild. Okay, and special thanks to Professor Roger Sloboda I hope I'm pronouncing that right. For giving me this guest lecture, and here are some more videos if you want to learn more about the subject. Thank you.